Hello, my name is Ross Macdonald. I'm the Chief Executive Officer of Cynata Therapeutics, and welcome to our company presentation at the Cell and Gene Meeting on the Mesa 2021. Cynata Therapeutics is an Australian stem cell and regenerative medicine company. First, please do refer to our safe harbour statement here and our filings on the Australian Securities Exchange platform and also our company website for further information about the company. We are, as I mentioned, an Australian stem cell and regenerative medicine company developing a proprietary platform technology which we call Cymerus for the manufacture of therapeutic MSCs for the treatment of serious diseases. Our unique manufacturing approach derives our product from induced pluripotent stem cells, or iPSCs. This approach overcomes all of the challenges associated with multi-donor derived manufacturing strategies. We've demonstrated strong safety and efficacy in both preclinical models of various diseases, as well as more recently, a clinical trial in graft versus host disease. We now have multiple clinical trials underway in a range of targets, one of which is in osteoarthritis, a phase 3 clinical trial being conducted here in Australia. Our targets are designed to address very large addressable therapeutic markets, particularly areas where treatment is inadequate or indeed completely ineffective. And given where our trading share price is at the moment, we have significant valuation upside. Why should we develop therapeutic MSC products? Well, it's becoming increasingly clear from the literature now that MSCs play a very important and central role in coordinating the body's mechanisms of defense, repair, and regeneration. Indeed, they've been described as the sensor and switcher of the immune system. The literature now shows multiple various means of MSCs influencing the immune system and switching on and switching off various components, as is illustrated in this cartoon. Such is the interest in MSCs. Now, as many of you will be aware, there have been more than 1,000 clinical trials registered in the past decade or so on clintrials.gov. What has become very, very clear in the past year or so has been a growing concern about the manufacturing strategies used to derive therapeutic MSC products. And the FDA's commentary late last year provided a very clear window to that agency's thinking about the existing strategies of MSC manufacture, and in particular, the reliance upon multiple donors for deriving MSC products, a reliance which ultimately results in inconsistencies, or as the FDA put it, functional heterogeneity between batches, which is absolutely a consequence of relying on different donors. Every donor is different, and so therefore cell products derived from those donors also will have different characteristics and therefore result in an inconsistency from batch to batch. Cynata's platform, on the other hand, addresses that challenge. We have no interdonor variability because we use an iPSC starting material derived from a single donation from a single donor, which allows us to essentially produce limitless quantities of identical MSCs. So this information from the FDA has been tremendously helpful for not only Cynata, but other companies to start to get an understanding of what the regulators are going to want to see when it comes to submitting BLAs and the CMC section of those dossiers. In summary, our platform, as I mentioned, utilizes induced pluripotent stem cells as a starting material. This technology is patented under patents held by both the company and also our licensor, the Wisconsin Alumni Research Foundation, which of course to this audience needs no introduction, as much of the groundbreaking work on stem cells, and in particular on iPSCs, was undertaken at the University of Wisconsin-Madison. So the reliance on iPSCs allows us an essentially limitless and identical starting material from which we can produce our finished MSC product. The advantages of an iPSC strategy is that we have a single blood donation from a single donor from which the iPSC was derived, that then allows considerable product versatility, manufacturing scalability, cost efficiency, and most importantly, from the regulatory perspective, product consistency. In summary, comparing the conventional process of MSC manufacture with Cynata's patented Cymerus process, you can see there in the top panel, the conventional processes rely on cells donated from multiple donors. Those cells are then purified, isolated, expanded in culture, and then utilized as the therapeutic product. Now, there's a couple of shortcomings in that process. The first is the reliance on multiple donors, the logistic challenge, and of course the inconsistencies between donors. But secondly, massive expansion in culture of MSCs has now been shown in the literature to result in a significant diminution of potency over population doubling or replications. This has been well established now in the literature. So the more the MSCs are expanded in culture, essentially the less potent they become. On the other hand, Cynata's Cymerus process relies on the iPSC starting material, which is essentially limitless in culture. We then, through our patented differentiation process, coax those iPSCs to become MSCs, and then ultimately that leads to our therapeutic product, which of course has characteristics of consistency and efficiency and allows us to produce high quantities of identical product. We've now leveraged that manufacturing platform into preclinical and ultimately clinical data. And our clinical trial in GVHD was a great success. In fact, it has been published as a front cover article last year in Nature Medicine magazine. And a quick summary of that data in patients with acute steroid-resistant graft-versus-host disease, which is a very serious condition and which over a 2-year period has got a typical mortality rate of in excess of 80%. At day 100, we saw a very good response in these patients. More than 50% had a complete response, that is a complete resolution of symptoms, and nearly all of the patients were still alive at the conclusion of the 100-day assessment period. We followed those patients out for a further, for 2 years in total, and we saw, again, very good efficacy results there with an overall survival of nearly 2/3 of the patients, compared with standard of care, where survival is less than 20%, or with other MSC products where at best survival is below 50%. So we were very encouraged by this excellent clinical trial and have now moved forward to a range of other clinical studies. We have a phase 3 in osteoarthritis underway in Australia at the moment, a phase 1/2 trial in Australia again in respiratory distress, primarily recruiting COVID-19 patients, but the recruitment criteria is much broader than that, allowing for patients, for example, with respiratory distress arising from influenza, for instance. Also, we're continuing our GVHD program that has now been licensed globally to Fujifilm, and it's anticipated that a phase 2 clinical trial will get underway as soon as the pandemic-related restrictions are resolved. The company is also investigating clinical trials in other indications, and we expect a trial in diabetic foot ulcers to commence later this year. Turning now to some more detailed descriptions of our clinical program, we have a phase 3 clinical trial underway in Australia in osteoarthritis that's funded by the Australian government. Osteoarthritis, of course, is a chronic degenerative disease that affects more than 30 million Americans. Market potential is in excess of $11 billion, and currently there is no cure. Treatment of osteoarthritis initially involves essentially analgesia, but as the disease progresses, ultimately patients end up with surgery, for example, joint replacement. So a product which will ultimately modify the outcome of this condition would be an outstanding result for MSCs and particularly for Cynata. That clinical trial commenced last year. It's now underway. We will follow the patients for 24 months after the first dose, and we expect results in 2024. We have a clinical trial underway in respiratory distress, which we call MEND. That trial is recruiting not only COVID patients, but also patients with other respiratory diseases who are admitted to an intensive care unit, primarily with respiratory distress. There's huge upside potential, and as we all know, respiratory distress was a major issue in medicine long before COVID came along. So even if vaccines ultimately result in a diminution of patient numbers with COVID, we still have a significant medical challenge in treating respiratory distress arising from other causes. That trial is now underway, recruiting patients, and we expect to complete recruitment later this year based on current trends here in Australia. We also expect to commence a clinical trial in diabetic foot ulcers. These are a particularly challenging chronic condition in patients primarily with diabetes. They're also called diabetic wounds. The market is very large, 400 million diabetics globally, with diabetic foot ulcers estimated to occur in up to 1/4 of those patients during their lifetime. Really, nothing has changed in the treatment of diabetic foot ulcers in around 100 years. So again, a therapeutic intervention which can ultimately result in accelerated recovery in these patients will be a great outcome. Our preclinical data was compelling, where we saw 86% skin restoration after 3 days following treatment with our Cymerus MSC product. Trial startup activities are now underway, and we hope to get that trial commencing recruitment later this year. It'll be in 30 patients, randomized, controlled trial, and as I said, getting underway later this year. The market opportunities of Cynata's broad and robust clinical trial portfolio are very attractive. Osteoarthritis is the major commercial driver, more than an $11 billion market opportunity. The respiratory distress and broader allied indications, also a very large market opportunity in excess of $8 billion. In addition to the clinical trials I've just described, Cynata has a broad showcase of preclinical data as well, derived from its Cymerus MSC products in areas like cardiovascular disease and also in transplantation biology and fibrotic lung conditions. We will look at commencing clinical studies either through partnerships or through other mechanisms as the company progresses. We see a number of pillars in our business strategy to derive shareholder value. And obviously, driving our clinical portfolio and generating robust, quality clinical data is a major value catalyst. But also we see opportunities for a platform technology such as ours to partner the technology with other biotech and pharma companies. And indeed, our partnership with Fujifilm is one such example of that. And finally, we also anticipate discussions with companies who might be interested in acquiring Cynata outright. And indeed, the company had discussions along those lines a couple of years ago with a number of potential acquirers, which ultimately didn't proceed, but nonetheless, we remain open to that as a business option. We see significant value upside potential for Cynata's share price, especially when we compare Cynata's market capitalization with some of the comparable companies in our space. And obviously, comparables are always challenged in deriving an apples to apples comparison. But nonetheless, here are 3 other companies that are involved in the cell therapy space and who use, in the case of 2 of them, an iPSC platform for the production of their particular type of cell therapy medicine. We have BlueRock, which was acquired a couple of years ago by Biotherapeutics for over $1 billion, or $1 billion valuation. Mesoblast, another Australian company, which has been a pioneer in the MSC space, market cap just under $1 billion US. And of course, the big guy in town, which is Fate Therapeutics, with multiple phase 1 clinical trials underway with a market cap of around $7 billion. So where Cynata is sitting at the moment with a market cap of around $60 million, I apologize, $60 million US, we see significant valuation upside potential for our stock. Looking ahead to catalysts for the company over the near and somewhat longer term, completing the startup activities and getting our diabetic foot ulcer trial underway is a major focus for us right now, and completing recruitment in our 24-patient men trial in respiratory distress this year will also be a major driver. We expect to complete recruitment and in the osteoarthritis trial in 2024, so that's still a way off, but obviously making progress along the way will be very important as well. We're looking to finalise a decision around the potential to conduct clinical trials in idiopathic pulmonary fibrosis and renal transplantation. And advancing our regulatory strategy in the US is also a major focus of our activities. We thus far have focused on Europe and Australia for our clinical programs, but we of course recognise the importance of progressing a regulatory strategy also in the United States. And that, as I said, is a major focus for our activities. And then finally, continuing our engagement with potential commercial partners for our platform technology, either by geography or by indication or disease area. These are discussions that are actively underway at the moment. Whether they will mature to a deal, of course, remains to be seen. But Cynata is working very hard to achieve that end. If we look now to a quick snapshot of our corporate profile, our stock price at the moment, as I mentioned, is relatively depressed, and we see a lot of upside there based on our portfolio and the likely catalysts upcoming. But at a market cap of around 77 million Australian dollars, which is around 50 million US dollars, we see a lot of value there yet to be realised. Our major shareholders are Fidelity International at 10%, Bioscience Managers, shade under that. Bioscience is one of Australia and Europe's largest and most sophisticated healthcare institutional investors, with a very successful track record. They cornerstoned a capital raising in the company about 9 months ago, coming in as a new shareholder following extensive due diligence. And our corporate partner, Fujifilm, is just under 6%. Our board and management are a highly experienced and well-credentialed team, particularly our chairman and Dr. Paul Watton. Paul is US-based and has considerable success, formerly the CEO of Cynata Therapeutics, which was acquired by Astellas, and now the CEO at Obsidian, which recently completed a very successful capital raise, private company. So very experienced team managing the company and looking to our shareholders' interests. I'd like to conclude now and thank you for your attention.